Hello, my name is Marisa and I'm a PhD student at the Open University in the UK, where I use Galaxy to analyse single-cell RNA sequencing data. Today we're going to go through this tutorial clustering 3K PBMCs with Seurat. So this is an introductory tutorial that's going to take us through one of the basic analyses that we'll do with single-cell data, where we're looking for clusters or groups of cells that had similar RNAs in them. The dataset we're analysing is this 3K PBMCs dataset, and that just means there's about 3,000 cells in it, and they are peripheral blood mononuclear cells. And Seurat is the pipeline or set of tools that we're going to use to perform the clustering. So if we head over to this other tab where I have Galaxy open, you can see I've got an empty history here. If you need to open up a new one, you can do so with that little plus sign at the top. And you can see here I'm working in the single cell lab on the European Galaxy server. I find that it's a little bit easier to locate the single cell tools when you're in the single cell lab, but you can follow this tutorial on the main server too. And another convenient thing when we're following a tutorial is if you open up tutorial mode. So if you click this little hat at the top, that will open up the Galaxy training network as a kind of an overlay in the same tab as your Galaxy. So this is the main page of the GTN. So we can scroll down to find the single cell topic. There it is, click on there. And now we've got the list of all the single cell tutorials. So we can scroll down to find the one that we're following. There it is. So you can see above it as well, there's another version of this tutorial where you're using ScanPy, which is another kind of popular single cell analysis pipeline. We want to follow the Seurat one today, so I'll click on there. Now we have our tutorial. So now you can kind of scroll down in here, you can see what steps you need to do and read all the material without leaving the tab that you have Galaxy in. The first thing we're going to do today is to upload the data that we're analysing. So these are our PBMCs. So when you see a black box like this on a Galaxy tutorial, you can usually hover over it and you'll get this copy option. So click on there to copy those file names. And then if we click outside of that tutorial, so on the background where we have Galaxy open, that will just close that down. So now we can head over here to upload, which is just there at the top left of the page. Click on that to open up the upload options. And what we want to look for is this grey button at the bottom, which says paste or fetch data. So click on there. And now we've got this little box here where we can paste in those file names. So then we press the start button and we should see those files appearing in our Galaxy history. There they are behind here. So now I can close down this upload interface. And here we can see we've got those 3 files, which are just the links that we copied from the tutorial. We've got one which is called genes, and that is the list of the genes that were analysed in the dataset. We've got a list with barcodes, and these are just the identifiers that were used for each of the cells in the dataset. And then we've got the matrix, and that is where all of the information about which genes or RNAs were found in each of those cells will be. So these are all grey at the moment because they're just waiting to run. They should soon turn orange, and that will mean the Galaxy is working on uploading these for us. And then once Galaxy is finished doing its work, they will turn green, and you'll see the same kind of pattern for all of the steps that we are running. So this could take a couple of minutes, so I'm just going to pause the video so that you don't have to wait too long to see what our next step should be. Okay, so my files have uploaded now, and if you head over to your history, once yours are ready, you can click on the name of the file, and that will just open it up in the history, a little bit of a preview. So here in the genes one, we can see the gene names. The first column there are the ensemble IDs, and then the second column are the gene names. That can be slightly kind of easier for us to remember and work with. You can see as well here the type of file. When we upload the files to Galaxy, Galaxy will kind of give its best guess as to what type of file they are. You can see here this is actually saying it's a tabular file. It should be a TSV file, but actually for the Seurat tools, I think we should be okay with a tabular file. So if we then click on the next one, the barcodes, you can see here you have the list of the barcodes. These are just the identifiers that were used for each of the cells. And you can see here Galaxy thinks this is a TXT file, which is not actually what we wanted it to be. So we can change this data type to the correct one. There's instructions for how to do this in the tutorial. Basically, by the name here, we can go to this little pencil, click on there, and that will open up the dataset attributes. If we go to the second tab along there where it says data types, click on there. Now we've got here the drop down box. You can see it's currently a text file. If we click on there, we can start typing in TSV, and we can see there's the option there. So I'll click on that, and then you do need to click save to make this actually change for you. So that file is now turned orange again because Galaxy's in the process of changing the file type for us. So while it's doing that, I'll click on the third file, the matrix. So this is where the actual information about which genes were found in our cells is stored. And you can see it's a particular type of file called a matrix market file. You can see it there in the preview. And this is basically a way of storing single cell data that means we don't have to store loads and loads of zeros. Because you can imagine if we're looking for 30,000 different genes in our cells, we've only got a very specific group of cells, blood cells in this case. So all of those genes that are expressed in other types of cells, like brain cells or muscle cells, are not going to be found in any of our cells. So there would be lots and lots of zeros where we didn't find any of those genes in any of our cells. And then even within kind of our blood cells, we've got different cell types in there. So some of them are going to be expressing a very specialized set of genes, and others are going to be expressing different blood cell genes. So there's going to be a lot of zeros in our data set, and storing all of that information would take up a lot more space than is necessary when we've got these kind of matrix market formats. So if you look at the little preview there, you can see on the first line, it tells us in the first column, that's the total number of genes that were in the data set. So that was our list of genes, just over 32,000. You can see there's 2,700 cells in the next column. So we had 2,700 cells in our list of barcodes. And you can see the third column there tells us how many non-zero values we have in our matrix. And the following lines after that are telling us for each gene, how many times we had how many, so the first column, so there, the 32,709, that is our 32,709th row or gene in the table. And you can see that in the second column that we're looking in cell number 1. And in the third column, it tells us for cell number 1 for that gene, we found 4 copies of that RNA. So we can see now, if I just close that down, there our barcodes file has turned green, which means it has now changed to TSV file format, which we can see there as well. So now that we have these 3 files, we can actually turn these into something called a Seurat object, which is just another type of single cell data type, which is the sort that the Seurat pipeline tools can interact with. So in order to do this, we can go back into tutorial mode again. So click that little hat, that'll open up the tutorial that we're working on. We can scroll down and we can see here it says creating a Seurat object. So the tool that we need is listed here in the hands-on section, the Seurat create tool. And when you're in tutorial mode, you should be able to click on the tool name there. And that should then open up the tool that we want to use here in Galaxy. So we are going to run the method create Seurat object, and we have a matrix market input as we discussed earlier. So in the first one, we need the counts matrix. That's our matrix, which is in there already. The list of gene names looks like Galaxy has put these in the wrong way around. So here I'm going to change this to genes.tsv for the gene names, and then the cell barcodes should be barcodes. So there's another couple of things to check here before we run it. So if we scroll down a little bit, we can see here we've got some filters, so we can filter out the really poor quality kind of cells that we had here. So I'm going to say if it include features detected in at least this many cells, I'm going to change that for 3. So I'm not interested in any genes that were only found in 1 or 2 cells, because I think that those are probably just likely to be mistakes in my data rather than genes that were really present. And then again, if it includes cells where at least this many features are detected, I'm going to put this to 200. So I think if there's a cell that was found that had less than 200 genes identified in it, I think that might not be a whole cell. It could be like a fragment of a cell that was processed in the experiment. So I don't want to kind of have that in my data sets to cause any problems. So I'm also going to leave calculate percentage of mitogenes as no. You can change this to yes here, which I would recommend doing when you're processing your own data, but we're just going to do that separately in this tutorial just so that we get a better idea of how we can use these tools. So now I can click Run Tool there at the bottom, and that will now create a new result in my history list. So this is this will be my new Seurat object, which is basically those three files that we uploaded converted into a new data type, which is kind of more convenient to use because it's all in one file and it's one that the Seurat tools can interact with. And you can see again that this is currently grey because we're waiting for it to run. And that is going to turn orange, hopefully fairly soon, and then green once it is completed. And then once we have created that Seurat object, we will then start working with it using the Seurat tool so we can do some quality checks and then we can start our clustering. All right, looks like this could take another minute, so I will pause the video. Oh, there we are, it's orange. So hopefully it won't take too long now. I'll just pause you and come back to you once that has turned green. Okay, so that didn't actually take too long for me. I've now got my output from the Seurat create step here. If I click on that name in the history, just like we did before, that will open up the preview. We can see here the data type is listed as RDS. That's fine. That's what we want to see there. And we can also go now to that little pencil icon again. So I'm going to click on there. This time I don't want to change the data type. I'm just going to change the name of this file. So instead of this kind of long description of the step that we did and the data sets that we included in it, I'm going to delete that and I'm just going to change this to input 3K PBMC. That'll just give me a more kind of recognizable name so that when we're using this data set in the next steps, or if I'm looking back at this history in a few days or weeks, I'll kind of have a better idea of what's in here. So then again, we click save and that should change the name in the history. There we go. So this is kind of, you don't need to rename everything in Galaxy, but it does kind of help you to remember and understand what you're doing and to kind of locate the right data sets. So if I head back into tutorial mode now with that little hat, we can scroll down and we can see the next step is to use this tool called Seurat Data Management. So again, I can click on here in the tutorial to open that up in Galaxy. So here, this is a tool that just lets us look at our data set because, if we click on this in the history, we can see there's not actually a little preview of it here. So if we actually want to find out a bit more about what's in this data set, we do need to use the data management tool to inspect it. And this is just because Galaxy can't sort of see inside the Seurat object in the same way that it could see inside those previous files. So I'm going to inspect this input 3K PBMC, and I'm just going to leave this as inspecting general information about it. So if I click run, we should now get another output, and this is just going to tell us what's in the data set. Obviously, we're going to hope that this is going to match up with what we know should be in there. So we're expecting to see those 2,700 cells and that 30-odd thousand genes in there, except that if you remember, we did do some filtering when we did that create Seurat object. So if there were any very low quality cells or genes that weren't present in very many cells, they won't have made it into this output that we are exploring. So we'll actually see how many of those cells or genes were kind of filtered out of the data set because of those settings that we chose. Okay, so that's green now, so I can actually click on that in the history to see that little preview of it. Or here I can also click on the eye icon next to the name in the history, and that will open up this file in the main kind of panel of Galaxy. So here we can see that the inspect general step is telling us that we have a Seurat object and that it has 13,714 features or genes in 2,700 samples or cells. So if we think back to what we saw previously when we looked at the files we uploaded, we've actually still got the same number of cells, 2,700. So it looks like the filter that we set to filter out cells that only had less than 200 genes in them hasn't actually filtered out any. So all of these cells have at least 200 genes in them. But we see that the number of features or genes has decreased quite a lot. So there were a lot of genes that we didn't find many RNAs for in these cells. So these could be genes that were found only in other cell types, like the brain or the muscle cells, or it could be just genes that we didn't find many of in this particular data set. So rather than kind of storing all of those empty zeros in our Seurat data set, that filter that we set for getting rid of genes that were present in less than 3 of our cells has kind of got rid of a lot of that unnecessary information. So now we have this Seurat object. Let me just close that down in the history. The Seurat object, which we have in number 4 here, this is what we're going to continue working with. And we now know what's in it because we've done this inspect the Seurat object step. So we've done some filtering when we created the Seurat object, but we do still want to make sure that we've got sort of good quality data in this data set. If there's any kind of cells that are looking a little bit strange or unusual, we might want to cut those out as well. So in order to understand which of our cells might be sort of poor quality, like fragments of cells and things like this that we need to get rid of, we're first going to calculate some more QC metrics, so quality control metrics that will help us to understand the cells in our data set. So if we open up tutorial mode again, we can scroll down past this inspect step to quality control. And you can see here, we're going back to the Seurat create tool in the next step. So I'll click on that to open it up. But this time, rather than doing the create Seurat objects method, I'll click on this drop down and I'll choose add QC metrics. So here we've got the file that we're using is that input 3K PBMC. And what we're going to do is match the pattern in gene names, which is already here. And we're going to match it with this thing, which is basically saying at the start, with that little kind of up sign, that's saying at the start of the gene name, look for this pattern. So that's MT hyphen. And this is a pattern that you will find in human genes that are present in the mitochondria rather than in nucleus. So we just want to count here how many of the RNAs that we found in each cell were mitochondrial RNAs rather than nuclear ones, because this is something that we can use to evaluate the quality of our cells. So I'm not going to change anything here because I'm happy with this. So I'll just go down and run the tool. And this is going to just add a little bit more information for each of our cells. So it's going to tell us what percentage of the RNAs that we found in that cell were mitochondrial RNAs. And that's something that can tell us kind of whether it's a good quality cell, because generally, if we have a really high proportion of mitochondrial cells, that can indicate that there was something wrong with the cell. Maybe it was dying and all its mitochondrial RNAs were being released. Maybe it was broken, so a lot of the nuclear RNAs that were floating around in the cytoplasm got leaked out of the cell, so it just kind of retained only the mitochondrial RNAs, and those can be signs that we need to eliminate those cells or filter out those cells from our data set because they're poor quality cells that are not kind of representative of what the kind of living or healthy cells should be. So we will calculate that kind of, as I said before as well, when we ran the create Seurat step to initially create that Seurat object, we did have the option of adding the mitochondrial metric at the same time, which would obviously have saved us from doing this extra step. I think in the tutorial, it's nice to kind of show these two steps separately. It's just so that you're aware that this add QC metrics option exists, because you can actually use that to add different types of QC metrics. For example, if you wanted to calculate the percentage of ribosomal genes, so those are kind of RNAs that are make up the ribosomes, because that's another thing that people sometimes need to do. So that is currently processing at the moment. And once it's finished, what we're going to do is create some pictures to show, different quality control metrics for all of our cells. So we can see if there are any cells that are kind of outliers, that have kind of extremely high mitochondrial percentages, or perhaps have a very small or high number of RNAs found in them. And that's going to tell us if there are any more cells that we should filter out of this data set before we do the clustering. Oh, there we go. It is now green. So we now have this number 6 in our history, which is the output of the adding QC metrics. So again, let's change the name of that just so that we know which one we're going to make the pictures with. So click on that little pencil and its name in the history. And I'm going to change the name of this. I'll just delete all of that and put in mitochondrial mitochondrial annotations. That's going to make it a lot easier to make sure I'm choosing the correct file when I'm going to use it to draw these pictures. So press save, change that name, there we go. So if we go back into our tutorial mode with that little hat, we can scroll down and see that the next thing we want to do is open up this tool called Seurat Visualize. So I can click on its name here, and now we can choose what sort of plots we want to make. So the first plot I'll make is a violin plot, which is already here, but I'm going to add on a couple of other different QC metrics to visualize. So I'm going to do N feature RNA, which is the number of genes that were identified in the cell. I'm also going to do N count underscore RNA, which is the number of RNAs that were found in each cell. And then I'm going to plot %.mt, which is that percentage of mitochondrial content that we just calculated for each of our cells. So if we scroll down a bit until we get to plot formatting options, click on there. I'm just going to say number of columns to display 3, and that will just make my plot look a bit more sensible, because those 3 plots will be plotted alongside each other. So you can either scroll down to the bottom to find the run tool, or because we don't need to change anything else, you'll also find that run tool button at the top here. So I can click on that, and that is going to draw some pictures for me. So while I'm waiting for that to run, I can actually start making the other plots that are listed in the tutorial. So back to tutorial mode with that hat, you can see there we've got 2 more sets of plots that we want to do, and these are going to be scatter plots. So if I click on that Seurat Visualize tool again, that will open it up. So again, I'm going to be using the mitochondrial annotations as the data set that I'm plotting. This time, rather than the violin plot, I'm going to click on this drop-down menu and choose scatter plot with feature scatter. So click on that. And this time I'm going to plot N count RNA, which is already in there. I'm going to delete what's in that second box, and I'm going to put %.mt in there, because that's what I want to plot against it. So now I can click Run Tool, and that should make that picture for me. And then I want to do one more scatter plot with N count RNA against N feature RNA. So actually, if I click on that number 8 in my history, which is the scatter plot we've just made, you can see here down at the bottom, there's this curly arrow. So I can click on there to open up the same tool with the same settings again. So you can see here it's already set to scatter plot. It's already got mitochondrial annotations as the file that we're going to plot. And all we need to change here is we want to change this so that it's N count RNA, which it already is. Rather than %mt, we're going to change this back actually to N feature RNA, because we're going to create a second scatter plot that has these 2 quality metrics plotted against each other. So now I can click Run Tool, and then we'll get a third plot in our history. So that little curly arrow can just be a very quick way to open up the same tool with the same settings, which, when you're doing the same step repeatedly with slightly different settings on it, that can be quicker. So we can see the violin plots are already here. So if I click on the eye next to them in the history, we can display those. It's kind of a big plot, so it can be a good idea to kind of right click or kind of double tap on this and select open image in a new tab. I can go to that tab and then see these 3 violin plots. So here we've basically just plotted for each cell what the level of each quality control metric was for it. So each of the dots in these plots is a cell, and kind of the violin shape behind it is just showing where most of the cells are found in this plot. So you can see here we don't have any particularly high values for most of the cells. They're all kind of clustered around near the bottom there, which is good. The first one here, N feature RNA, that's telling us how many unique genes were found in each cell. So if they're kind of particularly high or low numbers, those could be low quality cells. The N count RNA is the total number of RNAs found in each cell. So again, any particularly high numbers, that could indicate maybe instead of catching 1 cell, you caught 2. So you've actually got 2 kind of different cells in there. And then the percentage MT, as we discussed previously, if that was a really high, so there's a couple of dots up there up at the top, maybe those are kind of poor quality cells that we would want to filter out. So if we pop back into Galaxy, we can see those 2 Scatter plots are still running. They're just going to show us the same QC metrics in a slightly different way. It's kind of giving you the same information with kind of a bit more information about how the metrics relate to each other. And it's kind of, there's various different ways of plotting these QC metrics. It kind of depends which one you prefer to kind of interpret. So this one is finished now, so I'll click on that little eye. And again, I'm going to open this up in a new tab so it's easier to see. There it is. So this now is a scatter plot. Along the bottom, we've got the N count. So this is the total number of RNAs that were found in each cell. And going up the top here, we've got the percentage of mitochondrial genes. And you can see here we've got a few cells that had very high mitochondrial percentages, but very low total amounts of RNA. So those are probably going to be like fragments or cells that have lost most of their RNAs. They've been damaged. The mitochondrial RNAs are still in there because they were stuck inside the mitochondria initially. So that looks like poor quality cells that we want to get rid of, perhaps these ones here as well. There's also a couple of cells here that had very high total amounts of RNA. And again, those could be sort of these doublets or where we've captured more than one cell when we were doing the single cell experiment. We go back to the Galaxy tab. We're still waiting for this final plot to run. Once it's finished, we will then kind of decide where we want to set our thresholds for filtering out these poor quality cells, like what sort of percentage of mitochondrial DNA do we think is too high that indicates this damaged or dying cell, and which ones should we keep into our plot. And similarly, we can also filter for the number of genes that were found. So click on that eye, and again, I will open up that picture, a new tab. So here we can see now we've got the N count RNA, so that's the total number of RNAs found, and the N feature RNA, which is the number of unique genes that were found or unique RNAs that were found. You can see that obviously generally as one goes up, the other goes up. There's a few cells in here that have kind of very high numbers of both of these, and these are again likely to be not single cells because there's such kind of unusually high amounts of RNA and high amounts of genes found in them that, that's unlikely to be a single cell. So we'd kind of want to get rid of those, and we'd want to get rid of the ones that had the very, if we go back to this one, the very high percentages of mitochondrial RNAs. So if we go back to the Galaxy tab, let's pop back into tutorial mode with that little hat. And if we scroll down, we can see there's the plots that we were expecting to make, and they match the ones we've just seen, which is good. There's a couple of questions here that can kind of help you to interpret these plots. And then we want to set our thresholds. So kind of there's a question here which would be like, what sorts of thresholds would you set based on these plots? But the next hands-on step, we're going back to the Seurat Create tool. So I'll click on there again. And here we are back in that same tool, but this time the method we're going to select from this drop-down is filter cells by QC metrics. So I'll click on there, and now we've got various different options for what we want to filter out. So based on kind of what I've seen from those plots, I'm going to say I want the minimum N feature of RNAs. I'm going to set that to 200, so there should be at least 200 unique RNAs in each cell. But I don't want to have any more than 2,500 because I don't want those cells that had loads and loads of features in them, loads and loads of counts in them, because I think those might not be single cells. And then for my maximum percentage of mitochondrial genes, I'm going to set this at 5. That's kind of a very common threshold that you'll see in single cell experiments. It generally will work okay. I think based on our plots, that's going to get rid of kind of mostly the poor quality cells, but sometimes you'll want to set it a bit higher for certain cell types that are very energetic, like muscle cells, because they just naturally have a lot of more mitochondria in them. So let me just check that I'm filtering the right data set. Yes, mitochondrial annotations, because that's the one where we actually have these mitochondrial percentages in it. So now I will click Run Tool. And there we've got a new data set coming. So this is going to be our filtered data set where we will have got rid of some of those poor quality cells. So once that has finished running, we can revisualize it. So we'll run these 3 visualizations again, and we'll see kind of how those plots have changed. So since I know I'm going to do that, I'm going to get rid of these earlier plots so that we don't get confused. And we're just going to have a look, once this is done, we're going to see what we've actually changed in this data set by doing this filtering. So there we go, it's running now. I'll just pause this video while we're waiting for that to complete. Okay, so again, I'm going to click on the little pencil by this new output, and I'm just going to change the name of this to filtered dataset. So delete that and put in filtered dataset. This again is just helping me to keep track of what my datasets are and make sure that I'm using the right inputs for the following tools. So remember to click save to actually make the change happen. And we're just going to replot those 3 sets of plots that we made previously. So as I said, if we click on the previous ones that we did, so I'll click on that violin plot one, and I'll scroll down until I can see that curly arrow. If I click on that, that's going to open up the visualize tool again with exactly the same settings in it. All I'm going to do is change here the input file. So I'm going to use this one, the filtered dataset. So I'll click on there and then run the tool. And then for the next output that we did before the filtering, so that's number 8, click on that name, click on the curly arrow, and then click on the input file drop down and change that to the filtered data set. Then click run tool, and then we've got one more set of visualizations. That's another scatter plot there. So I'll click on that name, click on the curly arrow to rerun it, change the input to the filtered dataset, and click the button to run the tool. So we're just doing those 3 visualizations again. So we've now got 3 new outputs, but this time we're plotting the data after the filtering. So we'll be able to see what difference we made by doing filtering on those quality metrics. So hopefully we should see that we don't have any particularly high mitochondrial percentages, and we don't have those kind of outliers that had very high numbers of RNAs and features found in them. So that will just kind of confirm that our filtering has happened and that it has gotten rid of the low quality cells that we don't want to include in our clustering analysis. Okay, so this second round of plots is finished now. So again, I can click on the eye by the name of them in the history, and then open that up in a new tab to view it. So here are the new violin plots after the filtering. You can see kind of the difference most clearly for the mitochondrial percentage, because that axis now stops at 5 rather than I think it was 20 before. So those kind of really high mitochondrial percentages have now disappeared from this data set. Again, we can go back and we can look at the next plot, the scatter plot. So click on that eye. I'm going to open it in a new tab. There we go. So this again is the percentage of mitochondria has now been filtered at 5%. So anything that was higher than that has gone. And we also should see some differences in the other axis. Back here to the final third plot, that's the other scatter plot. I'll open that in another tab too. And here we go. And so again, those kind of really high outliers that were kind of up there somewhere have now been filtered out of the data set. So we've kind of got rid of the really extreme values that might be kind of damaged cells, dying cells, cells that were kind of more than one cell captured together. And now we're ready to analyze this data set because we think we've just got the kind of the high quality cells left, the ones that we actually want to cluster. So if we now go back into tutorial mode, so I'll click that, or let me just close down these tabs so they don't get in the way later. I'm going to click the hat to go back into tutorial mode. So here we can see again, kind of this is the steps we've already done. So those are the plots we just made. You can see kind of some of the differences in them if you examine them a bit more closely. Now that we're kind of happy that we have the good quality cells, we need to do a bit more pre-processing. So this is preparing our data set for analysis. And in the Seurat pipeline, there are two different options we can choose here. So we can either use the separate pre-processing tools. So these will do 3 different steps using 3 different tools. Or we can use this SC transform tool, which will do all 3 of those steps in one go. So there's a bit of discussion in the tutorial about why you might want to use SC transform in certain cases. But you can choose here when you get to the choose your own tutorial section, you can click on the option that you want to do. So you could click on SC transform to get the SC transform instructions, but I'm actually going to click on separate pre-processing steps. So I'm going to go through with the 3 separate pre-processing tools, just to kind of show you how that works. So depending on which route you'll take, you'll get slightly different results, just because we'll have prepared the data using slightly different kind of techniques. So whichever one you choose, you'll see kind of the results for it in the tutorial that you're following, as long as you click one of those buttons. So for the separate pre-processing steps, the first tool we will use is going to normalize the data. So if I click here on the name Seurat pre-processing, that will open up this Seurat pre-processing tool, and we're going to be working with the filtered data set. That's the one that we got rid of those low quality cells from. The method we'll use first is normalize data. So I'll leave that as it is. And the method we're going to use is log normalize. So I don't need to actually change anything here. I can just click run tool, and that is going to normalize the data for me. While we're waiting for that to run, we can actually set up the following steps. So if I go back to that little hat, we can see the next tool is going to be find variable features. So this is going to be finding the genes that had the biggest differences in expression across our data sets. They were expressed a lot in some cells, not much in others. So I'll click on the Seurat pre-processing tool again. That will open this up for me. So this time the input file is going to be this one that we've just normalized. So that's the one that's currently running. It's item 14 in the history. Rather than normalizing, we're now going to click on that drop down and choose identify highly variable genes with find variable features. So check the method is VST, that's the one we want to use. But these are just kind of different kind of techniques for identifying the genes. So if we scroll down here, we get to output list of most variable features. So I'm going to change that to yes, because I want to see what the most variable genes in this data set were. And I will leave that as a list of the top 10 most variable genes. So now I can click run tool. And the next step in pre-processing will be scaling the data. So again, we can go back into tutorial mode and we can click Seurat pre-processing again. And this time, the method used, we'll click on this and click scale and regressive scale data. While we're here, you can see in this drop-down menu, the SC transform option here. So if we had chosen that, that would have done all the normalization, the finding variable features in one go with a slightly different technique. So we're going to scale with scale data. And the input for this will now be the one where we found the variable features. That was number 15 in this history. And we're going to leave this as it is, except that we're going to choose for the features to scale. The default for Seurat is just to scale the highly variable features that we've just identified. But because it can make our plots look a little bit better, I'm going to click on that drop down and scale all of the features. So that's, something you might want to change depending what you're doing. So I'll scroll down and click run tool. So you can see here the normalization step has already turned green because that is finished. So that normalized the data. We're currently waiting for find variable features to run. So this will give us number 15 there will be the data set with the variable features identified in it. Number 16 there is going to be that list of the top 10 most variable features that we asked for it to output. And then once that is done, the final pre-processing step of scaling the data will be performed. So number 17 there is actually going to be the file that we go on to use in the following steps. So I'll just wait for that to run, and once it's done, I will come back and we'll take a look at our results. Okay, so this step is now finished, the find variable features. So if I click on the I next to the top variable features list output, then we'll see that list of the top 10 genes. So these are the genes whose expression kind of was most different across the data set. So we're kind of interested in these genes because the genes that are expressed at similar levels across all of our peripheral blood mononuclear cells are not really telling us much about the differences within this data set. But the genes that are expressed a lot by some cells and not much by other cells can tell us some information about what different types of cells we might have in this data set. So as well as looking at this list, we can also create a plot to see what our genes, our variable genes, look like. So this time, let's go into the Tools menu. So here on the left-hand side, if we click on that spanner, we can open up the search box for all of the tools on Galaxy. So if we search for Seurat Visualize, there we go, we'll see that. Click on that. If I click on that spanner again, it'll just close down the search for me. So now I'm going to draw a plot of my most variable genes. So I can use, I think in the tutorial it says to use the completed pre-processing scaled one. So we do need to wait for that to finish. So I'll put in that number 17 is the data set we're going to plot because that has the scales data in it, or it will do once it's finished. And for our method use, the sort of plot we're going to draw, I'll click on that drop down. And if we look in here, we should see plots variable genes with variable feature plot. So I'll click on there. And now we could scroll down. I'm going to say label the top most variable features. I'll change that to yes. And again, I'm going to show 10 of them. So those 10 genes that we just saw in the list will now be labeled on this plot. And it's just going to be a plot showing us the variance that was kind of calculated for each of these genes. So we'll see, how much more variable these top 10 genes were than the rest of the data set. So I will click Run Tool. You can see my scale data set step is now processing. Once that has finished and that data set is ready to be used, then my plot is going to be created. So while I'm waiting for that to happen, I'll just pause the video and then I'll come back to you once my plot is ready. Okay, so my plot is now ready. I'll click on the I next to its name, and then I'm just going to open this in another tab again. And here we can see we've got the plot of how variable the genes were. Our top 10 most variable genes that we saw in the list previously are labeled. You can see they're obviously a lot higher up in this plot than most of the genes. You can see the red genes here are our 2000 most variable genes. 2000 is just kind of the default number that's identified by Seurat. I think the number is different if you're using the SC transform version. But basically, these are the genes that we're most interested in because they were the most different between our cells. So if I close down that tab, head back to the main one. So let's open up tutorial mode again with that little hat and then scroll down. That's the gene plot. So dimensionality reduction, this is the next step we want to do. So we have these genes that varied across our data set, but there's still 2000 of them. That's quite a lot of information. It's kind of too much for our brains to make sense of or for us to kind of draw useful plots. So dimensionality reduction is going to look for groups of genes that kind of varied in a similar way, and it's going to identify those as principal components, because what sort of dimensionality reduction we're doing is principal component analysis. So it's a bit more of an explanation of this in the tutorial to kind of help you try and get your head around what that means. But let's just go on to the hands-on step. So we'll click here. So we're going to open the tool Seurat Run Dimensional Reduction. I can do that by clicking on the name here. And oh, I forgot to rename this file. So that's file number 17 there. After we finish the final pre-processing step, I'll just click on the pencil next to its name, and let's rename this as pre-processed data. This is just, I would still be able to find this file if we didn't do that, because this data history isn't that complicated, but it's just kind of a good habit to get into, really, because, if I come back in the future, I'll be able to kind of very quickly spot that in my list of data sets if I need it for something. So again, let's open up that tutorial mode, click on Seurat Run Dimensional Reduction to open the tool. And here you go, you can see now I'm running it on pre-processed data. That's obviously a lot clearer when you're, clicking on this drop down and selecting which data set to run on. Having these kind of renamed ones does make that a lot easier. So we're running it on pre-processed data. The method we're choosing is run PCA, dimensionality reduction. And then let's check what else we might want to change. If we scroll down, we'll leave this the same. We're just going to change here the output the list of the top genes. So let's change that to yes, because we're kind of trying to understand in this tutorial what we're doing. This is going to tell us the genes that were kind of had the strongest impact in these principal components for the kind of the top principal components. So we'll output the list of top genes. We'll leave this as print for 5 PCs. So these are the kind of the strongest principal components. Print for each of those the top 5 genes that had the most impact on that principal component. So now we can click to run the tool. And you can see here again, we're going to have 2 outputs from that tool. So the first one is the Seurat object, which has the PCA results in it. And the second one will be this list of the top genes associated with our top 5 PCs. So if that doesn't make too much sense to you, you should read through the tutorial, because it does kind of explain what I'm talking about here when I'm talking about PCs and the top genes in them. So hopefully that will make a bit more sense to you after you've kind of read through that and looked. After this, we're going to visualize the results of this PCA and kind of show you different ways to kind of understand how the variable genes and the top genes in these PCs kind of relate to our data set and kind of the principal components that we're calculating. So I think this could take a little while to get started, so I'll just pause the video and come back to you once it has finished. Good, so that has finished. We have got here, as I said, kind of the second output here. We can click on the I for, and that is our list of our top 5 PCs. So this is PC1, PC2. And for each of these, we've got the list of the top 5 genes that had the most influence on that PC, so both in a positive way and in a negative way. So these are the genes that kind of at one end of this axis, the positive genes were being expressed, at the other end of the axis, the negative genes were being expressed. And again, this is explained more in the tutorial if you want to kind of understand what this actually means. So the other output here that we had, this one, number 19, we can't view that because that is a Seurat object that has all of our PCA information in it. I'm going to, again, rename that. So clicking the little pencil icon. And then I'm going to call this PCA results, just so that it will be easy for me to work with later on. And then click Save to change that name. There we go. So the tutorial will kind of guide us through various visualizations of these PCA results, kind of relating them to these genes that we found as the top positive and negative genes for the different PCs. And I think this will hopefully help you to kind of understand the meaning of what we're doing. So if we click on the little hat for tutorial mode, we can go down to the first visualization we want to do. So first we're going to do a dimensional loadings visualization. So click on Seurat Visualize to open up that tool. And we're going to visualize the PCA results. That's correct. In the method used, we will change that to this one, a visualize reduction results with visdim loadings. Click on there. So I'm going to reduce this so we don't have too much information to overwhelm us. The number of dimensions to display, let's change that to just 3 of them. And then for each of those, the number of genes to display, we'll leave that at 30. Again, I think we want to scroll down kind of as we did earlier, plot formatting options. Let's click on there and change the number of columns to display to 3, so that we've just got each of our 3 dimensions plotted alongside each other. We do also want to change the size of the plot. This is just going to make it look a bit better. So I'll change that to yes. And I'm going to change the width of the plot in pixels to 3000. This is just kind of what's recommended in the tutorial. And that's because we know this is going to be a plot that's going to look a bit better if we change the size of it. So it's also quite useful to know that these sorts of options are available when you're using these tools yourself. So now I will click Run Tool, and that's going to create a plot that's going to show me these kind of loadings, or how strong of an influence each of the genes had on the principal components. So what we'll expect to see is, if I click back on the I for the print PCs output, these for the PC1 plot that we're just making, we're going to see these top positive genes and the top negative genes on there, and the top positive genes will have very high kind of positive loadings, and the top negative ones will have very large negative loadings. So we're plotting these top 3 PCs, and we're plotting kind of the impact, or the strength of the impact that each of these genes had on those principal components. So while we're waiting for that to be created, we can keep working our way down the tutorials through more of the visualizations of these PCA results. So this time I'm going to click on the Seurat visualize that we've just created in the history. I'm going to get that little rerun arrow again to open up Seurat visualize. So for the next plot, we're going to plot again the PCA results. But for the method used, I'll click on that drop down, and I'm going to choose a visualize dimensional reduction with dim plot. So this is going to show us the top kind of dimensions in a scatter plot for the PCA analysis. So the only other thing I need to change is the name of the reduction to use. Rather than a UMAP, we have a PCA. So there we go. And then, oh, I did not need to change the size of the plot for this one. You won't need to do this if you haven't rerun it. So I'll just change that to no. And I think again, yes, I think that should be fine. So yeah, you won't need to. That should already be no if you opened Seurat Visualize again from tutorial mode or from the tool list. But because I reran it from the one where he changed that, that was kind of already changed. So here we go. Dim plot of the PCA results. So I'll run that tool. So that's the next visualization that we're going to have. We also had another couple of visualizations to do. We can visualize feature plots, which will basically be the same as that dim plot, scatter plot, but with the expression levels of different genes colored in them. So let's open up tutorial mode, and we can see what I mean. So you're getting a preview here of what the plots are going to look like. Those are the VisDim plots. The dim plot will be that scatter plot. And the next one we're going to do is here we're going to do the feature plots. So the reason I've opened up tutorial mode is because I know I'm going to need this list of genes. So here I'm going to highlight that and copy them. And then we're going to open up Seurat Visualize. So I will click on that tool name. So here again, we're still visualizing the PCA results. But this time in our method used, I'll click on there and I will choose, where are we? Down here, visualize expression with feature plot. So I can click there. We now have the option of features to plot, and that list of genes that I just copied from the tutorial, I will paste in there. So you might recognize these names from, these are the top positive and negative genes for PCs 1 to 3. So those are kind of the most extreme genes on each dimension, positive and negative. So we're going to plot PC1 on the x-axis, PC2 on the y-axis. We'll change the name of the reduction to use again to PCA because we're plotting PCA reduction UMAP. And this time we are going to actually change the size of the plot again. So I will click on that no, change it to yes. And I'm just going to say make the plot a bit wider. So I'm going to put it to 3,100 pixels wide. And this is because I'm plotting multiple genes, so I know there's going to be a few different plots in this picture. So let's click to run that tool. And then actually on this one again, so this is the feature plot. I will click on that in the history, click on the rerun arrow. And I'm going to actually do this again, leaving everything the same, except that on our dimensions to plot, I'll change the x-axis one to PC2, just put in the number 2, and the y-axis to PC3, so just put in the number 3 in that box. Leave everything else the same, and click Run Tool. So now that is going to kind of plot the kind of the most important genes for our PCs on our PCA plot. I think we're still waiting for all of these to run. So if we continue with that little hat down tutorial mode, we'll see we've just done these 2 feature plots that we're still waiting for, which are going to look like this. And then we're going to do some more visualization with heat maps to kind of show, again, the expression of the most important genes for each of our PCs. So we're going to do 2 different versions of the heat maps here. So the first one, I'll click on the Seurat Visualize tool again. So still visualizing the PCA results. You know, these are kind of like various different visualizations that show you different things. You wouldn't need to necessarily make all of these plots when you're doing your own analysis. They're just kind of different ways of understanding and checking what's going on with your data. So for the method used, this time I will choose visualize reduction results with dim heat map. And I'm going to go in the dimensions to plot. I'm going to delete this so that it's only PC number 1 in there. And I'm going to scroll down then to advanced options, click on there. I'm just going to plot the top 500 cells so that we don't have too big of a plot. And I'm also going to change here, this little button, I'll click on that so it changes to yes, so that it will return an equal number of genes with positive and negative scores, because I want to see both the positive and negative genes for the PC in my plot. So then I will click Run Tool, and that's going to do a heat map with just the top genes for the first PC. So if we click on that one that you've just done, the heat map, click on the rerun arrow, and then we can do the same thing, but we can just change here the dimensions to plot. So if I put, instead of just plotting 1, I'll put 1:15, and that will plot each of the first the top kind of 15 PCs, and we'll get the same sort of plot, but for each of those PCs. So this time we will change the size of the plot, because that's going to be 15 different plots all in one page. So let's increase the width of the overall page to 4,100 and the height to 4,100. And then we can run that and wait for our results to appear. Just click there to close that down. You can see some of our visualizations have now appeared. So this was the first one that we made, the VisDim loadings. I can click on the eye to view that. And again, I'm just going to right click on that and open it in a new tab so it's easier to see. So here we can see this was where we plotted for the first 3 PCs the loadings of the top positive and negative genes for them. So here you can see for PC1, we've only got sort of 1 negative gene, so this is the negative side of the axis. That was that top negative gene for it. And then we have all these kind of positive ones on the positive side of the axis. So this top gene here, CST3, that will be the top gene that we plotted in the feature plot, and it will also be in that list of genes that we printed out from the run PCA stat. And you've got the same kind of plots for the next 2 PCs, so these kind of the strongest kind of axes or PCs that had explained our data, and you can see which genes had the biggest positive and negative impacts on each of those PCs. So I'll close that tab. Let's look at the next visualization. So this is the dim plot. I'll click on the eye and again open this in a new tab. Here we can see it's kind of like a scatter plot where the axes are the first and the second principal components. Each of the dots is a cell, and you can see they're kind of spread out, some of them kind of forming sort of groups on here. Like these are not clusters yet, we haven't done our clustering. This is just showing that PC1 explains quite a lot of the variation, because a lot of the cells are at one end of the axis and a lot of them are at the other end, and PC2 also, they're quite well spread out along this axis. So this is kind of showing that our PCA analysis seems to be working. If I close that down, we can see the next 2 plots are those feature plots where we plotted the top genes associated with the top 3 PCs. So if I click on the eye of that first one there and open that in a new tab, you can see this is the same slightly squashed now the same as the previous dim plot that we saw, but we've coloured it in now by the expression of these top positive and negative genes. So you can see the top row here is the top positive gene for PC1. You can see that's mostly expressed at the positive end of the PC1 axis, and similarly the top negative gene is mostly expressed at the kind of negative end of the PC1 axis. And similarly for PC2, its top positive gene is expressed mostly at the positive end of that axis. Its top negative gene is it's a little less of an obvious relationship there. And you can see for these top genes from PC3, they're not really as associated with the axes. Like I may have, hold on. Yeah, I think I misinterpreted that plot because it appeared in a different order than I was expecting. I think if I look back at my one of my feature plots, if I click that rerun arrow, I think in the plot formatting options, I should have changed I should have put the number of columns to display as 3 before I made this, and it would have the order of the plots would have made a bit more sense. Let me just quickly run that for you, and we'll see if it appears. So yeah, the I was misinterpreting these plots because I was expecting them to be in a different order. So the CST3, that's the top positive gene, which I was correct about for PC1, so it's mostly at the positive end. But actually, the CD79A is not the negative gene for that one. It's actually the top positive gene for PC2. So it's at the positive end of the PC2 axis. And then this one, the third plot here, HLA DQA1, that is actually the top positive gene for PC3. So it's not really related to the PC1 or the PC2 axes. The MALAT1, actually, again, it doesn't show a huge relation, but this is actually the top negative gene for PC1. So it's, to a certain extent, you can see it's a darker purple color kind of at that negative end of the axis. I think that PC just didn't have any particularly strong negative associations with it. NKG3, that's the top negative gene for PC2. So you can see it's at the negative end of that vertical PC2 axis. And PPBP is the top negative gene for PC3, which obviously is not plotted on these plots because this is PC1 by PC2. So it's not kind of related to either of those axes. So then if I go back to my Galaxy tab and we look at the next feature plot, again, these plots are going to be in a slightly weird order because I forgot to change the number of columns. So if I open this in a new tab, so here again, we can see this is now the horizontal axis is PC2, the vertical axis is PC3. We can see here the top positive gene associated with PC1 doesn't have any association with the PC2 or PC3 axes. CD79A, which was the top positive gene associated with PC2, is now at the positive end of that PC2 axis. And HLA DQA1, we can now see an association between it and the PC3 axes. It's kind of slightly hard to see in this picture, but it is mostly expressed in those cells above the 0 there on that axis. MALAT1, that's associated with PC1, shows no real association with either of these axes, as we would expect. NKG7, so this is the top negative gene associated with PC2. There it is at the negative end of that axis. And PPBP, now you can see it's got a negative association with the PC3 axis. So these plots are kind of just explaining to you how these genes relate to the principal components that they kind of influenced. So hopefully if you kind of read through the tutorial, that will make sense to you. And hopefully if you remember to change the number of columns for those feature plots, yours will look a little bit different. We can see, there we go, there's the one I just reran here. If I click on that and open it up in a new tab, this is what I was expecting to see, where you have the because you put them in 3 columns, you've got the 3 positive genes for each axis on the top and the 3 negative ones on the bottom. So those 2 are the ones associated with PC1. So that's kind of, I think that's a slightly clearer arrangement of them, which is why I've got the in the tutorial it advises you to put them, to change it to be in 3 columns, but it's still the same plots. So we've looked at the two sets of feature plots there. We've then got the heat map. So if I click on the eye of that first heat map that we made, I'll open that in a new tab. So this is showing the expression of the top kind of genes in the top cells just for PC1. So you can see that MALAT1, which was that top negative gene, you can see in these purple cells, there's very low expression of it. There's high expression in these other cells. And then you can see these kind of blocks, the yellow and purple blocks. It's showing that there are a set of genes that in some cells are highly expressed and in some cells are expressed at very low levels. And this is basically saying, our PC axis is explaining a lot of the variation in the data because there are these two groups of cells that some cells express them very highly and other cells express them at very low levels. So again, if we go back to our history, we can click on the eye for the next one. And then we can open this in a new tab, and this should be the basically 15 heat maps for each of the top 15 PCs. And you can see we have these very strong associations for the top PCs, because these are the PCs that explain most of the variation in the data. And kind of as we go down through the PCs, you can see by the time we get to the end, there's still some kind of pattern there, because they explain some variation, but there's obviously not such a strong pattern as in those kind of first PCs. So there's kind of more information about how you interpret these plots in the tutorial. So you can read through that if you want to understand a bit more about what's going on. But there's one more plot that we can make, and this is a plot that's just going to help us decide how many of these PCs we actually want to use in the next part of our analysis. Because as we just saw in those 15 plots, some of them were had a much stronger influence than the others. So if we go into the tools menu, and I'm going to search for Seurat Visualize, click on there, click on the spanner again to close that search. So again, we're still visualizing the PCA results because we haven't changed that Seurat object yet. The method used, I'm going to look for determine dimensionality with elbow plot. Click on there. So I'm going to plot the top 30 dimensions. I know we calculated 50, but I'm just going to plot 30 because from looking at those 15 PCs, we could see that by the time we got to the bottom of those, they weren't having a massive impact on the data. So PCA reduction is correct. I just need to press Run Tool. And this elbow plot is going to help me decide which of those PCs are explaining the most about the data and how many of them I should use for my actual clustering in the next steps. So this is kind of a fairly useful plot for deciding that. It's basically just going to plot kind of how much of the variation in the data was explained by each PC, so I can choose the ones that explain a lot of the data and not bother including the ones that didn't kind of add much extra information about my data set. All right, so I will, that's gone orange now, so it might be fairly quick to finish. And then we'll take a look at that and decide how many PCs to use. Okay, so that's finished. I'll click on the eye to view that. And then again, I'm going to just open this in a new tab. So now I can see it clearly. You can see here, this is called an elbow plot because there's this bend in it. And what we're looking for is where there's kind of a sharp bend and where it kind of reaches close to this bottom axis and becomes kind of a straight line along here. You can see there's kind of a big drop between the first PC and the second PC and the third PC. These are kind of fairly large drops because each of these is explaining a large amount of the variation in the data. And then once you get to here, there's only tiny little drops between them. So each additional PC is not really adding much more information about the data set. So we don't really need to bother calculating using these for our clustering. So some people might choose slightly different kind of cutoff points here, depending, on how they prefer to do it. I'm going to say we'll use the top 10 PCs. That seems to be around about where this corner happens, and it's a nice round number. So let's close this plot and go back here. I'll click that little hat to open tutorial mode. We'll have to scroll down past these heat maps and the elbow map. To see the next step, which is actually where we're doing the clustering. So there's two steps to this. First, we calculate a neighborhood graph, and then we look for clusters within that. So here's the hands-on step. We're going to use a tool called Seurat Find Clusters. So I can click on the name of that. Here we go. The input will be, again, this PCA results. And we are going to do the step of compute nearest neighbours with find neighbours. We're working with that PCA reduction. And as we just decided from the elbow plot, we're going to use 10 dimensions for the input. That's actually the default that's already in here. So we don't actually need to change anything in this tool. We can just click Run Tool and wait for that neighbourhood graph to be created. This is just kind of, it's basically working out which cells were most similar to each other. And for the next step, we're going to cluster that neighbourhood graph up into groups of cells that were similar or close to each other. So we can actually click on that Find Clusters in the history, use that rerun arrow to open up the Seurat Find Clusters tool again. This time the input will be our neighbours, our output from Find Neighbours. And we are going to change the method now to identify cell clusters with Find Clusters. I'll change the resolution from 0.8 to 0.5, and that's again just because that's what the tutorial recommends. And the resolution is basically going to decide kind of how detailed our clusters are, like how many clusters it's going to be broken into. And we know kind of from the tutorial that this is a good resolution for this data set. We're leaving this algorithm as original Louvain. There's like various different algorithms that you can use to work out where your clusters should be split up in your neighborhood graph. And, you can experiment with those if you're analyzing your own data. For the tutorial, we'll stick with this one. So I'll click Run Tool, and that will start running once the previous step that it's going to use as its input has completed. So this will actually be kind of the clustering step. It will have identified the clusters. We won't actually be able to see anything in these results because that's the rat objects. So once those are finished, we're going to do another dimensional reduction step. If I open up tutorial mode, we can scroll down. So we've done this step, we've done the fine clustering step. The next step will be this one, running a non-linear dimensional reduction. So this is just going to give us a slightly different kind of way of visualizing the data that can be a little bit clearer when we're looking at clusters. So we're actually going to take the option of running a UMAP dimensional reduction. So let's get that running now while we're waiting for the clustering steps to finish. So I can click here on the name Seurat Run Dimensional Reduction to open up that tool. This is the same one we used previously to do Run PCA. This time we're using as the input this Find Clusters, which we're still waiting to run. For the method used, we're going to run UMAP, because this is the sort of picture we want to take a look at. We're going to base this on the PCA reduction. So the PCA reduction is going to be used to create this UMAP reduction. We will leave this as it is. We are running it on dimensions. So we're going to use 10 dimensions as the input. So again, we don't actually need to change anything here. So let's just click Run Tool. You can see there the Find Neighbours is finished. We're still waiting for the Find Clusters to run. And then once that has completed, this UMAP step can be run too. So let me just pause the video while we wait for those to run. Okay, so that step took a while for me, but it's finished now. I'm just going to go to this Run UMAP results, click that little pencil and its name in the history, and then I will change its name here to UMAP results. Again, that's just to help me find it later on. I press save to confirm the change. There we go. So we've now done a kind of second type of dimensionality reduction. This is similar to the PCA, but the reason why we're doing this UMAP as well is it's going to give us kind of better pictures when we're looking at our clusters. So it's just going to make interpreting these results a little bit easier. So let's do a visualization to see kind of how this UMAP plot with the clusters on it compares to kind of what we saw previously with the PCA. I'll go to the tool search here on the left-hand side, and I go to search. Again, we're looking for the Seurat Visualize tools. So I'll click there to open that up. Click on that spanner again just to hide the tool search. And now the input that we're going to plot is our UMAP results. And for this first plot, I'm going to choose as the method used visualize dimensional reduction with dim plots. So I click there and just make sure that the name of the reduction to use is UMAP, which is what we now want to plot. So I can click Run Tool to get those results. And this is going to be very similar to the first the kind of dim plot we did of the PCA results. It's obviously going to look a bit different because this time we're plotting our UMAP. And we should also see it coloured in based on the clusters that we've just identified. So while we're waiting for that, I will open up the tutorial mode and I'll scroll down here. So this is the step that we've just done, the first visualization. We're going to do another visualization step, but this time we're again going to plot the genes, the top genes associated with the first 3 PCs. So I'm just going to copy this list of gene names again. And then I will open up the Seurat Visualize tool by clicking on its name here in the tutorial overlay. There we go, I've opened it up. So again, I'm plotting the UMAP results. This time I'm going for visualize expression with feature plot. So I click there. The features to plot in this box, I'll just copy in that list of genes that I just copied from the tutorial. So again, another thing else to change, we're plotting, I do need to change the number of columns to display. I think this is what I forgot to do last time. So in the plot formatting options, if I just click there, the options will appear. Then I can scroll down until I see number of columns to display, and I will put 3 in here. That's just going to put the plots in the order that I kind of want them to be in. I will also click here on change size of plot. I'm going to change that to yes, and I'm going to change the width of the plot to 3100 pixels so that all of my 3 plots will fit in nicely. So now I can click Run Tool and wait for that plot to appear. So this might again take a little while, so I'll just pause the video while I'm waiting for those plots to be made. Okay, so I'm just going to start the next step while we're waiting for these results to appear, just to save us some time. So I'll pop into the tools search, and we're actually doing another visualization. So we want Seurat Visualize again. Click on that and then click on the spanner to close down the search. So we're still visualizing the UMAP results, and we do actually want this one, the violin plot, because this is just going to be another way to show those genes that we just plotted on the feature plot. So in the list of features to plot, I'll delete the default in there and I'll copy in that list of genes again. That's just the ones from the tutorial. And again, I want to go to the plot formatting options and select the number of columns to display as 3. And then I will also change the size of the plot. So down at the bottom here, I'll change that to a yes. And I'm going to put, again, the width of the plot as 3100 and then run the tool. So while I'm waiting for that to run, let's take a look at these previous 2 visualizations that we did. So the first one here, the dim plot, I can click on the eye by its name, and then I'm just going to open that image up in a new tab. And here we can see this is our UMAP plot, and it is coloured in by the clusters that we have just identified. So these are the groups of cells that had similar RNAs in them. And we can see from the key that there are 9 clusters, because the clusters always start with cluster 0, so that's actually 9 of them. These cells are plotted as a UMAP plot, so these are kind of the UMAP axes that are similar to the PC axes we saw before. So you can see we've got some pretty good big groups of cells that are kind of separated out from each other, and they've been separated kind of within those groups into different clusters. And now if we go back to Galaxy and we click the eye on the feature plot, this is where we coloured in those clusters based on the top genes associated with our top 3 PCs. So again, I'll open this image in a new tab. There it is. So here we can see this is our same UMAP plot, and I've just coloured them in by the top genes associated with the PCs. So whereas previously when we looked at these genes on the PCA plot, they were associated with the axes, you can see now that they're no longer associated with these axes because these are now the UMAP axes, but we can see that there are groups of cells that have kind of higher or lower expression of some of these genes, and that kind of makes sense to us because this UMAP plot and the clusters that we identified, we did that based on our PCA. So it makes sense that the genes that kind of had a big influence on the PCA results would have some sort of connection with our clusters and our UMAP plot. And if we pop back here, this violin plot that we're just waiting for now is going to show these same PC genes. There it is. If I click on the eye next to that, and then I can open that up. This is just another way of visualizing the same information. So here we have violin plots where each of these violins on each plot are our clusters, so clusters 0 to 8, and we've coloured in kind of the expression of those top genes. So the dots here again are the cells, and the kind of the violins show where most of the cells are found. So we can see that, you know, in the second plot along, the CD79A gene was kind of mostly being expressed by cluster number 3. So that's just kind of, you know, some people might prefer to see the violin plot, some people might prefer to kind of look at these kind of UMAP plots. It kind of gives us the same information. So if I close down those plots and head back to Galaxy, we can now see what our next step is. So now we're going to actually try and find marker genes. There's various different tools and ways of doing this with Seurat. So the next section of the tutorial just kind of quickly shows us different ways of finding markers. And essentially a marker is just a gene that was significantly more expressed by one group of cells than another. So rather than kind of using those genes associated with the top PCs, we can now look at within the clusters that we've identified as what makes that cluster different from the rest of the cells. So if I click the little hat to open up tutorial mode, we can scroll down past these visualizations that we just created to the Find Marker Gene section. So the first option that we're going to try out is this one. We're going to use Find All Markers. So this is going to find the markers in each cluster that were significantly more expressed by that cluster than any of the other groups of cells. So I'll click on the tool there, which is Seurat Find Clusters, and that will open up here behind. So the input we're going to use is that UMAP results, and we are going to change the method to identify marker genes with Find All Markers. So I click on that. We're going to use the default method, which is Wilcox. Again, there's kind of various options and methods that are available through Seurat for kind of more advanced users. So we're going to we're going to actually go into the advanced options. So I click on advanced options to open these up. And you're going to scroll down until we see only return positive markers. So I'll click that to change it to a yes. And now I will run that tool. And basically, when we look for positive markers, we're looking for genes that are more expressed in the cluster we're interested in, rather than ones that are expressed less often in that cluster, which would be like negative markers. So we can choose to kind of see both the positive and the negative markers, or just the positive ones, as we've done here. And you can see that this tool produces two outputs. The first one is kind of an RDS file, and the second one is the list of the markers, which will give us some statistics and the names of the genes that it found were associated with each cluster. So I will just pause the video while we're waiting for that to run, and then I'll come back when we can take a look at the results. Okay, so this is taking a really long time to start running for me. So while I'm waiting for that to happen, I'm going to move on to the next one of these find markers from the tutorial. So this one we have started, and we're hoping it will finish at some point. The next one they suggest trying is if we just want to find the markers of one specific cluster. So in this case, we can rerun the same tool. So let's click on one of these outputs that we're waiting for and hit that little curly rerun arrow. So this is going to open up the Seurat Find Clusters tool again. So we are using as the input the UMAP results, but in this case, we are going to choose as the method used, instead of this find all markers, which will look at all of the clusters, we're going to click on that drop down and choose identify marker genes for specific groups with find markers. So if I click on there, we'll get slightly different options for what we want to compare. So I'm going to choose compare markers between clusters of cells. I'm going to choose yes there. It's going to say which class do you want to define the markers for. So I'm going to say I'm going to look at cluster 2. So in this case, what I'm going to ask this tool to do is to look for the markers that define cluster 2. So what genes were expressed more by cluster 2 or less by cluster 2 than in other genes. So because I rerun this, I think I'm going to need to go into the advanced options and just check that, yeah, it is going to identify positive and negative markers for me. So that is fine. I don't need to change that. So identify with find markers, compare markers between clusters of cells, and identify markers for cluster 2. So that is now ready to run, so I can scroll down to the bottom and hit that run tool. That's going to give me another 2 outputs. That will be again the RDS file and the list of the markers. And while we're waiting for all of these to run, let's move on and run the next tool. So we are going to use find clusters again. So let me click on that in the history and hit that rerun arrow. So this time we're going to actually, oh, that was actually running. So I think the previous one has maybe just got stuck and I probably need to rerun that again, which I'll do in a second. I'll finish my thought and continue with the next one, which is where we're going to use find markers again to compare specific clusters. So rather than comparing cluster 2 to all of the other clusters, we can compare one cluster to one other cluster or to multiple clusters. So you'll see what I mean when I fill this in. So we're using the Seurat Find Clusters tool again. Our input is the UMAP results. And we're using identify marker genes for specific groups with find markers. So again, we're still comparing markers between clusters of cells. So we want it to have yes here. In this case, we're going to actually, rather than cluster 2, let's look at cluster 5 because that's what's in the tutorial. So we're going to compare the markers of cluster 5, not compared to all of the different clusters that are present. We're compared to cluster 0 and 3. So we'll put those in with a comma between them, and then we will run this tool. So this will tell us which genes were found more often in cluster 5 than in clusters 0 and 3. And there's one more option that we can use here to find clusters. I mean, there's many other options, but these are the ones that were presented in the tutorial. There's loads of different options that are available here if you want to kind of explore the different sorts of comparisons you can make. So here, again, we're in the find clusters. Our input is the UMAP results. We're identifying marker genes for specific groups with find markers. And we are, again, comparing markers between clusters of cells, because these are the clusters we've just identified. This is what we're interested in at the moment. We're going to define the markers now. So I'm going to get rid of that 5, and I'm going to look at cluster 0. And I'm going to delete these, so I don't want to compare them to specific clusters. But I am going to scroll down here. I'm going to change the minimum log fold difference to test. So this is how much of a difference I'm looking for. I'm going to increase that from 0.1 to 0.25. Just following what the tutorial says here. You know, if you're interested in what all of these parameters mean, you can obviously go into it in great detail. Then I'm going to change this time. I'm going to change where it's a select test to run. Instead of Wilcox, I will click on that drop down and I'm going to select this one, which is ROC or ROC. And I'm going to, this is just kind of a different way, a different statistical test that you can use to compare expression between clusters. And in the advanced options, I'll click on there and I'm going to again choose only return positive markers. And then I can just click run tool and get the outputs from this test. And in this case, this is just showing that there are different tests available. This one, the ROC test, will test the classification power of the genes. So this is kind of going to give us a score for the genes between 0 and 1, where 0 means that gene doesn't help us to kind of identify the cells that should be in that cluster, and 1 means that gene is kind of only really expressed by cells within that cluster and not by any of the other genes in our data set. So you can see here most of these are running. That first one that I started running is still grey. That's quite unusual. I mean, generally when something isn't working on Galaxy, you shouldn't rerun it because you're just kind of adding more jobs into the queue and kind of causing a problem. But seeing as I've been waiting for a really long time for these to run and the following ones are running, I'm going to assume that this could just be some sort of glitch. So I'm actually going to press the rerun arrow on that job and just not change anything because I want to do the same one again. So I'll just click run tool and see whether that makes any difference. Because you see here, one of my following jobs has finished. So that is the new, those 2 at the top now will be the job that I've just rerun because it's still stuck on grey and hasn't even started running down here. So in the meantime, here's one of the following ones that we did. I think if we click the rerun arrow on this, we can check which one this is. So this is the find markers where we were comparing cluster 5 to cluster 0 and 3. So if we look as an example at the markers list from that output by clicking the eye, we can see the sorts of results that we'll get from these markers tests. So here you have a list of the gene names in column 1, just p-values and the average change in that gene. So in this case, it will be the fold change will be in cluster 5 compared to clusters 0 and 3, and we can see the adjusted p-values. So this is telling us these are the genes that were expressed most differently in cluster 5 when compared just to cluster 0 and 3. And for all of these other tests that we've done, you will see results that kind of are for the similar comparisons, depending on what we ran. So, for example, this one, number 38 in my history, this was from where we ran the find markers just on our cluster 2. So if I click on the eye for that, we will see a list. These are the genes that were significantly differently expressed in cluster 2 compared to all of the other clusters in our data set. So we can see that cluster 2, if we look at the average fold change there, those are kind of positive. That's a positive value, an increase in expression of IL-32. So we can say that cluster 2 is expressing more of IL-32 than the other clusters. So we're waiting for, there we go, see that one that wasn't running before has now turned orange. So it is actually being processed now by Galaxy, which is good. So probably we'll see if that ever actually finishes or starts running when I check on it in the future. Occasionally these things will happen, but yeah, I wouldn't generally recommend rerunning things that you're waiting to run or that are orange and just taking a long time, because if you just keep repeating and rerunning steps, then you are kind of adding more work into the servers and kind of contributing to the problem if things are running slowly. But in this case, it looks like that did just get stuck and hasn't been running. So these kind of once their results come, we'll see again similar kind of find markers lists, which will kind of show us the genes that were kind of most significantly different based on these different tests that we were running. They'll look slightly different depending on what test we performed and kind of what clusters we were looking at, but they're all kind of interpreted in fairly similar ways. So there's all these different kind of tests that we can do to look for marker genes. There's kind of, comparing specific clusters, comparing all the clusters. But kind of one of the reasons why we'd really want to do this would be to identify genes that can tell us what the differences are between our clusters. So in this case, we have all of these peripheral blood mononuclear cells, and we are kind of interested in whether our clusters match up with kind of known cell types that should be present within that group. So what we will do next, if we click into tutorial mode, we can scroll down past all these different find markers that we've tried out, and we can look at this section, identifying cell types. So the first thing we will do is we're actually going to just rerun the find all markers test, but with slightly different settings. So I'll click on Seurat find clusters. That will open up the tool here. So we are going to run this again on the UMap. So I need to go past all of these different find clusters and click on that UMap results as my input. And we're going to change the method to identify marker genes with find all markers. So we're comparing which genes were expressed differently in each cluster as compared to the rest of the data set. So we will change the minimum log fold difference from 0.1 to 1.0 because we want to focus just on the genes that had the biggest differences between clusters. We're going to leave this as the Wilcox method, and we will say we'll limit the output. This is our list of markers. I'm going to click yes to limit the number of markers for each cluster that I'll get in that list, and I'll leave that as a limit of 10. And then in advanced options, if I just click on there, I can scroll down and find where it says only return positive markers. So I'm going to click on that and change it to yes, because I really want to know what are the top 10 genes for each cluster that were expressed more in that cluster than in the rest of the data set. And I'm going to hope that those are going to tell me what sorts of cells are in each of my clusters. So let's click Run Tool. And there we've got these two find all markers outputs, which we're going to wait for here. So while we're waiting for that to hopefully this time start running and complete running, we can look back here. This is the output of one of our find markers. So this is where we use the slightly different test. If I click on the I in the markers list output in the history, you can see the results. And here, this is these are the values that are telling us which of the genes were kind of had the highest classification power for cluster 0, I think it was. So we can see here that the highest one is this one, RPS12, and that had kind of this high value of 0.827, and that is kind of the most, has the best, that was kind of the gene that told us that was most expressed in that cluster compared to the other clusters. So we're just waiting for those. So that was the one that we reran, and then the next two, 45 and 46. That's the test that I've just run to identify the markers that had the highest fold changes of over 1, which are the genes that I'm going to use to identify my clusters. Let's just scroll down. Yep, still grey down there. So hopefully these ones will finish soon, and then we can move on to the next step in identifying our cell types. So I'll just pause you again while that's running, and hopefully this time you won't be paused for too long. Okay, so that one at least has finished. Let's click on the I for that. This is actually very similar to the results we'll eventually get for that first find all markers that we tried to run. So here we have the list of genes that were associated with each cluster. Here we've got 10, we've limited the results here to the top 10 genes for each cluster. So you can see there are 10 here that are associated with cluster 0. So these are 10 genes that, because we limited this to positive genes that had a fold change of over 1, these are all kind of genes that were more expressed in cluster 0 than in the other clusters. And similarly, then we've got the same for cluster 1, cluster 2, and on until the end of all of our clusters. Now you can see that one finally finished. So one way that we could identify our what sorts of cells are in each of our clusters would be to Google these genes or to search for them in kind of databases and try and work out what sorts of cells might be represented by each of these clusters. Another option would be to take kind of what's known as a supervised approach, where we take genes that we know should be expressed in specific cell types that we expect to see in our data sets, so different types of blood cells, and we would then look at compare their expression between the clusters. So if we open up tutorial mode with that little hat, we can scroll down parts here. So here you can see the known list of markers for PBMCs. So these are kind of the genes that we know should be expressed in specific types of blood cells. And we can then visualize our data set in different ways to see where these genes are being expressed. So if we click here on this hands-on step of Seurat Visualize to open up, oh, first, before I close this down, I'm going to copy these two gene names, which are two genes that are known to be expressed in B cells, which are a type of blood cell. So now I'll click on the Seurat Visualize tool to open that up. We're going to, before I do this, I should rename that output, because we have a lot of find markers and find all markers outputs, and we want to make sure that we are actually using the right one when we're doing all of these visualizations. So what I will do is rename that final output. So I'm looking for the markers list here, which is output number 46 from that final find all markers that we run. I'll click the little pencil in its name, and then I will change the name of this to DE, or differentially expressed markers, just so that I know which one of these outputs to use, and then I'll press save to confirm that. There we go, the name is changed. So if we pop back into our tutorial mode, click on Seurat Visualize again, and we're going to we're actually using the UMAP results, so I didn't need to do that renaming right away, but it's good to do it while I was there, because we are going to use that list later on. So we have the UMAP results as our input, and we're going to do a violin plot, so that's correct. And then in the features to plot, I will delete that default in there and copy in those two gene names that I just got from the tutorial. So now I will click Run Tool, and we'll now see some plots that show which of our clusters were expressing those two genes the most. So these are the two genes that we expect to see in B cell markers. So if we see that one of our clusters is expressing a lot of these genes, we will think that those must be B cells. So if we just go back into, let's just have a look at that DE markers list of results again. We'll click the I. So we could, as I was saying, we could also kind of look through these genes to try and work out which types of cells we have in these clusters. And you can see here for cluster 3, the top two differentially expressed markers that were present in that cluster but not in the others are CD79A and MS4A1. And these are genes that if we kind of search them up in a database, we wouldn't see that they are associated with B cells. So these are actually the two genes that we're just plotting in that violin plot. So obviously we've seen here in our list of the differentially expressed genes that those are associated with cluster 3. So in our violin plot, we will expect to see that most of the cells expressing these two genes are in that cluster. So that's kind of two different ways that you could be identifying your genes. You could either look at your list of differentially expressed genes and search them up in databases, or Google them just to see what types of cells might be expressing them. Or you can start off with that list like we saw in the tutorial, if I pop back into tutorial mode. So this list of known genes that kind of previous research has told us is associated with specific cell types like those B cells. And kind of, you might use a combination of both approaches. You might choose one or the other, kind of depending on, what you're looking at and what sort of research you're doing. If I scroll down, we can see that we can do the same kind of thing for other types of cells. So here the tutorial suggests we do this second violin plot, but this time for T cell markers. So these are other types of blood cells. There's a few different marker genes here. So if I copy those, 4 different ones, and then I can click on Seurat to visualize. So again, we want to change the inputs back to the UMAP results, but we are still doing a violin plot. To features to plot, delete what's in there and copy in those gene names. So these are the ones that are in different types of T cells, because we've got a few extra genes here. We're doing 4 genes this time. We are going to just change in the plot formatting options. Let's change the number of columns to display to 4. This is just going to make our plot look a bit better. And then scroll down to change size of plot. Change that to yes. And I'm just going to make the plot a bit wider so that we can fit in those 4 different sets of violins. And I'm going to change the width of the plot to 3100 and then click Run Tool. So once these are finished, we're going to have one set of violin plots that tells us which clusters we're expressing those genes that are known to be in B cells, and one set that's going to show us which clusters we're expressing genes that we know to be in T cells. Okay, so those have now finished plotting. So I'm going to go to this first violin plot, the B cell one that we made, click the eye next to it. Then I'll open that up in a new tab so we can take a good look at it. So here we can see, as we expected, cluster 3 is basically the only cluster that has much expression of either of these genes, and kind of that's as we expected, because when we looked at our find markers results, we saw that these 2 genes, which are the B cell genes, were markers of this cluster. So head back to Galaxy and let's look at the next violin plots. These are the T cell markers. So again, I will open that up in a new tab. Here we can see we've got 4 groups of T cell markers. CD8A is very much associated with that one cluster, cluster 4, but the others is kind of less obvious what sorts of T cells we're looking at. IL-7R is actually a gene that we can use to identify to identify kind of T cells in general. So we can see that kind of cluster 0, 2, 4, and perhaps 7 are kind of T cells. And then these other genes, the other 3 genes are actually expressed by certain types of T cells more than others. So kind of we would have to use all of this information in combination to identify groups or clusters of different types of T cells. So I'm not going to explain too much about that now. There is a bit more information about how you could use these different markers to identify different types of T cells in the tutorial. But we're not kind of just looking at B cells and T cells. There are other types of blood cells that we expect to see in our data. So if I open up tutorial mode by pressing on that little hat, then if we scroll down past the T cell markers that we've just done, we can see that we can actually colour in our plots by a whole load of different genes that are associated with different types of blood cells. So these are actually all of the genes that were in that table up here, which had all of the different types of blood cells that might be in our data set. So you can see some of them are expressed by more than one of these canonical marker genes. Some of them you're only looking for one gene in this list. So if we go back down here, I'm going to now copy this list of genes, and then let's open up the Seurat Visualize tool again. We can do this by clicking on Seurat Visualize. So we again want to change the input back to the UMAP results. And then this time, let's make some feature plots. So this is kind of similar to what we did before, where we were looking at the top genes from the PC. So visualize expression with feature plot. But this time, rather than looking at our top PC genes, we're actually going to plot here in features to plot. I'm going to copy in that list of known marker genes for different types of blood cells. Scroll down, check the name of reduction to use is UMAP. That's what we want to plot. And then scroll down to change size of plot and change that to yes. And we're going to make the plot 4,100 pixels wide and then 4,100 pixels high, because we're plotting quite a lot of these feature plots with different genes. So now let's click Run Tool and wait for that to happen. While we're waiting, we can also do some more violin plots. That's the next step in the tutorial. And this is just kind of showing that there are 2 different plots here that you can use to identify which clusters are expressing these canonical markers. So let's click on that Seurat Visualize, click the rerun arrow to open up the tool again. So we want us to input the UMAP results. We want to change the method used to having a scrolling issue there. Change the method to violin plot, and then for the features to plot, delete that default, and copy in again that list of all of the marker genes. So again, let's make sure, so change size of plot. So yes, we want to leave this as the larger size that we set previously. And then click Run Tool. So this is going to give us two ways of visualizing which of the clusters we're expressing which of these known marker genes. And based on that table that's in the tutorial, we should then be able to identify what types of cells were present in each of our clusters. So let me just pause you while those are running, and then we'll come back and take a look at the results. Okay, so since these are finished, let's go to that feature plot and click on the I next to the name. And then I'm going to open this in a new tab so we can look at it. Here we can see this is our UMAP plots. This is the same as when we were looking at our clusters before, but this time each plot is coloured in by one of those canonical markers. So we could look through this plot and we could compare it to our previous UMAP where we showed where each cluster was located. So if we scroll down, we can see if we can find that plot. Still grey there. So here we had the, after our UMAP, we had that dim plot. If I click on the eye there, we can open that in a new tab. So this should be our UMAP coloured in by cluster. So you can see there, for example, that group down there, coloured in green, that's our cluster 3. And if we compare that to which of the marker genes are being expressed in that cluster here on these feature plots, we can see there that MS4A1, which is that known marker of B cells, is mainly being expressed in that cluster 3. So we could then say those are our B cells. And we could do that based on all of these different marker genes in all of our clusters. We can actually work out which cell types are present in which clusters. Alternatively, if we go back up to the top here and click the eye to look at this violin plot, open that in a new tab. We could do the same thing with these violin plots. So for each marker, again, there's that B cell marker expressed mostly in cluster 3. So we can look through these and work out which cell types are present in which cluster. And kind of either one of these plot types can work. It just kind of depends which one you prefer. So let's close down these, go back to Galaxy. So if you kind of follow the tutorial, I'll click on that hat for tutorial mode. If you follow the tutorial, it'll tell you a bit more about how to identify your clusters based on these markers, and we can then actually rename our clusters. So rather than having like cluster 0 or cluster 3, we can actually give them names of the cell types that we have identified. So here I'm going to copy this list of names. And then I will click here on the tool Seurat Data Management to open that up. And I'm going to put in as the input, again, my UMAP results. I'm going to change the method used to manipulate Seurat object. And I'm going to choose as the manipulation to perform. I'll just click on that drop down and I'm going to choose rename idents. So idents are basically the identities that we've given to cells, which in this case are the cluster numbers. So I want to rename all of the idents, and for the new names, I will just copy in that list that we got from the tutorial. So these are the cell types that we have identified by looking at which of those known canonical marker genes were expressed in each cluster based on those plots we just made. So I will choose save copy of old idents first, change that to yes. So that just means that it will also store the cluster numbers just in case I need those again in the future. So now I can click Run Tool. That's going to give me a new Seurat object, which has the cell names and not just the cluster numbers stored in it, because that's going to be a lot kind of easier for me to work with if I'm kind of analysing this data set in the future. Like rather than having to remember that cluster 3 is my B cells, I can just see, oh, those are B cells. So we can also then draw a picture of this to show that the change has happened. So if I click on the spanner to open up the tool search, let's open up Seurat Visualized again. There it is. I'll click on that spanner to close the search. So now we're going to be drawing this latest data set where we've changed the names. That's currently number 51 because it's still running. I will rename that once it has finished running. So this data set we will draw. We're going to choose as the method used the dim plot. So this is the UMAP plot that we made before, where we had the clusters on it. So check the name of reduction is the UMAP, which is this one. And because we've changed the names, we've changed the ident from the cluster numbers to the names of the cell types that we just identified. This should actually give us the same plot, but with the cell type names in it. So I'll click to run the tool. And then I will just pause the video while I'm waiting for those two steps to complete. Okay, so let's click on the eye for that plot we just made, and then open it up in a new tab. Here we can see this is our same UMAP plot with our same clusters, but now we have renamed those cluster numbers into the cell types that we identified. Making it a lot easier to kind of interpret our plots and to understand what sorts of cells we're looking at. So we've actually managed to kind of cluster our single cell data and to identify kind of a biological meaning behind those clusters because they're expressing markers of different cell types that are known to be part of this data set or expected to be part of this data set. If I just close this down, there are two kind of additional steps in the tutorial. If I click on that hat to go back into it, the tutorial mode, I can scroll down past there where we've made our UMAP plot. And you can see there's a couple of other plots here that you can make to kind of understand your data. So we're going to do two different heat maps. First one, let's copy this list of genes. So these are these canonical markers of different cell types that we've just used to identify the data, the clusters. So I can click copy to copy that. And then let's click behind this tutorial mode to go back into Galaxy. I'm going to then go again to this upload data that we used at the beginning. Here you can see those files I pasted in earlier. And I'm going to click again on paste fetch data. And in this box, I will copy in that list of genes from the tutorial. Let's change here from auto detect. Click on that drop down and we're going to search for tabular. Click on that because we're going to basically create a table that is this list of the canonical markers. So now click start. We should see this appear. There it is in our history. You can close down the upload tab now. So this is basically going to create a tabular data set, a table where each row is one of these canonical markers that we have just used, and that is going to let us use this list of genes in the following plot that we want to make. So once that has happened, let's check back on tutorial mode. So we are now creating this list, which we'll rename once it's done, and check the data type. And then we're going to visualize a heat map where we use this data set as the list of features to plot. So creating this table kind of lets us do this by using a file in our history rather than kind of typing in the list of all the features. So while I'm waiting for that to upload, I'm just going to pause the recording again. So that is still processing, but while we're waiting, I did forget to rename this data set, the output of the Seurat data management. This was where we renamed the clusters into the cell names. So I'll click the little pencil by the name of that in the history, and then I'm going to change its name to annotated clusters. And again, this is just so that we know which data set we want to use when we are now doing our plots and things later on. So I can click save to make that change. There you can see it's changed. and this pasted entry has now turned green. So if I click on that in the history to get that little preview, we can see that it is indeed tabular, so that's the correct data type, and we can see that list of genes that we pasted in are available there. So I'll click on the pencil as well for this one, and I'm going to change the name of this list to canonical markers. And that's just going to help me to make sure I'm choosing the right data sets to use in my plots. So let's pop back into the tutorial mode by pressing on the little hat. And you can see that we're going to use again that Seurat Visualize tool. So let's click on the tool name there to open it up. So we are going to use as the input file the annotated clusters. So these are the clusters that we had renamed using our cell types. The type of plot we're going to do will change the method used to visualize expression with do heat map. There are a couple of heat maps here, so make sure you choose the right one. We want this one, do heat map. Click on there. And now we can choose as our list of features to plot. We can click on the drop down and select, if we scroll up, the canonical markers. So this is this list that we just pasted in. If I click on there, and we can then just make sure the formatting is going to be okay. So on the plot formatting options, click on there to open it up. We're going to look for the size of text above color bar, above color bar. There it is. So we're going to reduce that to 3.5 just to make sure that everything fits in okay. And then the angle of text above the color bar. We scroll down. Let's change that. We can drag this along up to 60 degrees. That's just going to make sure again that everything fits into the plot. If we scroll down further, change size of plot, change that to yes, and we're going to make the plot 4,100 pixels wide and 4,100 pixels high. So now we can click Run Tool, and that's going to give us a heat map showing the expression of those canonical markers. And while we're waiting for that, we can also make work on the final plot that we want to make. Let me just click on that canonical markers to close it down. So we're going to do another heat map where instead of plotting the expression of those canonical markers, we will plot the expression of the most differentially expressed genes that we identified when we ran find all markers. So if we scroll down to look for that file that we renamed as DE markers, so this is the output of the final find all markers that we ran. Let's just click on that name. And we can see here that the data type is CSV. We actually want to have a tabular form of this that we can use as the list of genes for another heat map. So let's click on the pencil next to the name of this file. And we're going to go to the second tab along where it says data types. Click on there. And we're going to go down here to where it says convert to data type. So we're going to make sure that what it says here is tabular using convert CSV to tabular because we want to change the CSV file into a tabular file. So then we can click the Create Datasets button. And that, if we scroll back up to the top of our history, there we go. We've created now a new output DE markers, which is just waiting to run now. And that is going to be a tabular version of that list of the differentially expressed genes for each of our clusters. Remember, that was a list that we'd limited to the top 10 differentially expressed genes for each cluster. So once that is ready, we're going to do a couple more things to make this output usable, because at the moment it has all of those statistics in it, but we really, for our heat map, we just want the column that has the list of the gene names in it. So once that's finished, we're going to actually cut out that list of the gene names, and then we're going to use that as the list of genes for our heat map. So it's a little bit convoluted, but sometimes you just have to kind of change your data set a little bit to get it into the form that you can use to kind of create a plot or do a particular type of job on it. So that has now finished. If we go to this new version of DE markers, click on its name in the history, We'll see that now in this preview, it's saying the format is tabular because we've changed it. And you can see in that little preview there that we have that list of gene names that we want to use for plotting in column 1, plus all of this other statistical information in the additional columns that we don't actually need. So what we want to do now is just to cut out this column of gene names. So if we go into the tool search by clicking on that spanner, let's search for a tool called cut. So there we go, this top one here that we want to just cut columns from a table. Click on there to open it up, and then click on that spanner to shut down the tool search again. Give us a bit more space to work in. So we want to just cut out column 1. So in this first box, let's get rid of C2. So C1 is column 1. That's all that we actually want to keep. And it's delimited by tab because this is a tabular format. And then the input we want is this new tabular version of DE markers. So now we can run the tool, and that's going to just cut off that first column that we can then use as an input for visualization. So while we're waiting for that to happen, let's go back and look at this other heat map that we created. So we can click on the eye to do that. So this is a heat map showing the expression of the canonical markers in our clusters that have been renamed according to cell type. Let's open that in a new tab and take a look at it. So here you can see for each of our canonical marker genes here, which are down the side, we can see the expression levels in the different clusters that are kind of along the top here. So for example there, if we look at our B cell cluster, if we look down there, we can see that yellow block there is for that gene that is known to be expressed in B cells. That's MS4A1. And you can see that, there is some expression of kind of other genes in that in that cluster. There's kind of some other clusters that have some cells expressing this B cell gene, but generally there's a pretty clear link between different canonical markers and different cell types. So let's pop back into Galaxy. That is now running, so hopefully that won't take too long, and then we'll have our list of just the gene names from our find markers, our differential expression test. And we'll basically make another heat map that is using those genes as the input instead of the canonical markers. We can kind of take a little look and see what the differences are. And we have the heat map, we have kind of the feature maps, the feature plots that we did, and the violin plots. These are all kind of different ways of visualizing which of your clusters are expressing which genes. And you can either be looking at canonical markers, or you can be looking at the most differentially expressed genes based on your statistical tests. And these are all just different ways of kind of understanding your data and kind of working out what your clusters actually represent. And in this case, we're kind of quite happy with our clustering because we can see that our clusters are expressing genes that are known to be associated with specific cell types, and that those are cell types that we expect to see in this peripheral blood cell data set. Okay, so if we click now on that output, the cut on data 55 in our history, we can see that we now just have that one column of the gene names that we've cut out from our DE markers. So I'm going to click on the pencil to change the name of this data set. So rather than cut on data, let's call this the input DE markers. And that will just make sure that we use this input for our list of genes for the heat map. So click Save to confirm that change. Now let's just go back to where we had the previous heat map, because we're going to use all the same settings. So click on that in the history to open it up. Click on that rerun arrow to open the tool again. So here we're back in Seurat Visualize. Our input again is still the annotated clusters. We're using do heat map. The one thing we want to change is the list of features to plot. So rather than this canonical markers, let's click on that drop down and choose the input DE markers. That's the one single column of the gene names that we've just cut out of our results. So click on that. This is why kind of renaming it is useful, because it lets us very quickly be sure that we're using the right data set there. So we're going to check the plot formatting options. Yes, our size of text above the color bar is 3.5. Angle of text above color bar is 60. And also check that the size of the plot is still 4100 by 4100. And then click Run the Tool. And this is just going to give us another heat map at the top of our history, which will show, again, as for the previous one, it will show the expression of this list of genes in each of our clusters. And that's, again, it's just a different way of visualizing and a different way of kind of understanding our data. So that will be the final step that we need to do in this tutorial. Okay, so that one is now ready to view. So I'll click on the eye next to its name there, and then open that picture up in a new tab and take a look at it. So here we can see again, we've got a heat map. We've got now this kind of long list. So these are our top 10 differentially expressed genes for each cluster. So these were the kind of the positively associated genes. And then we've got along the top here, the different clusters. So again, we can see we have for our B cell cluster there, we've got this block of yellow, which is the highly expressed genes, and these are the top differentially expressed genes associated with that cluster. So again, this is kind of a different way of viewing your data. You know, it can be nicer to look at a picture like this rather than just at that list of gene names. And you've got the different options. You've got these heat maps, you've got the violin plots, you've got the feature plots. They're all just kind of different ways of viewing your data. So now we have finished this whole tutorial. Let me just click on that little hat again to scroll down to the bottom here. At the bottom of the tutorial, you'll find various links, so you can take a look at the workflow. So that's kind of the tools that we just used. There's also some suggestions for other tutorials you might be interested in. So one thing you could do, you could go back and if you did these separate pre-processing steps like I did here, maybe you want to try the SC transform tool or the other way around. There's also, as I said at the beginning, the ScanPy version of this tutorial. And if you go to the list of single cell tutorials on the GTN, you'll find there's also the case study series of tutorials, which will let you perform clustering and some other apply some other analyses to a slightly kind of messier data set than the one we've been using here, which is kind of a very nice beginner's data set for this introductory tutorial. So if you get to the bottom of the tutorial, it's nice if you click this button to say that you've finished, just to let kind of the people who work at the GTN know that we've been making content that you actually have found useful. And there's also at the bottom here a list of kind of places where you can find help if you get stuck or if you have questions about using Galaxy. So thank you very much for listening.